sections, and we have four different organs actually. Okay, today we will go through those gastrointestinal system, upper digestive system organs, and starting with the uppermost element, which will be the mouth, and inside the mouth we will see the tongue itself. Just to make a close look in the lowest magnification. Okay. For the closest, highest, lowest, lowest magnification, I will try to show you the overall structure in the tongue. As you remember, in all the digestive system organs, we were trying to identify four basic layers comprising the organs. So starting with the uppermost layer, which will be the mucosa, and then a thin layer of lamina propria, and broad one should be the submucosa, and then going through those submucosa, and then going through the muscle tissue here, muscularis externa. But we call in the tongue, it is directly straight muscle elements are filling the muscularis externa, so that's why we do not name directly muscularis externa. And we are more interested in the mucosal surface, so here we go to the next magnification. And if you see, okay, all right, okay, thank you. Now, as you see, here we are observing the tunicum mucosa. It is formed with the first of all the epithelium, and then the lamina propria in blue color here. So we are still observing the tunicum mucosa, but the first sublayer would be the epithelium itself. So we should recognize that the epithelium here at the tip of the arrow is a stratified, and the uppermost layer is the squamous, so stratified squamous epithelium, and as you can easily recognize, the basal cells having round nuclei, but the uppermost cells having the spindle shape of nuclei, showing us the squamous type. So it is a stratified squamous epithelium. And the lamina propria, having more connective tissue elements, is stained with the blue color. Since our preparation was stained with the malloresin staining during the histological preparation, the connective tissue elements are stained in blue. But what about the nuclei? They stain in red color, so we can easily understand the epithelial cells. Okay, going through the lamina propria and then the submucosa, still connective tissue elements, and then some connective tissue bundles, as well as some blood vessel sections. Blood vessels could be some arterioles with thicker walls, or veins or venules, let me say, with thinner walls, okay? And still they are filling up the submucosa. And the next layer is the muscularis layer. As you see, the muscularis layer is clearly visible with the striated muscle cells. So the striated muscle cells giving functional characteristics of the tongue, very motile tongue, we have the straight muscle cells in different orientation of section. Some of the straight muscles are cut through cross sections, or some of them are in longitudinal sections. You can always try to make bigger magnifications, and by the help of light adjustment, it is always good to see the striations. Okay, I am trying to make some some magnification all through those fiber muscle fibers. I am trying to show you. Some striations, but okay, it is difficult. You see long fibers of straight muscles showing some striations. Maybe, yeah, okay. Maybe here, some of them. But it's clearly different from the smooth muscle, which we will see in the next section. But going through the mucosal surface once more, we will be coming to an interesting feature of the tongue itself. It is, I am going through the, too much. I'm still going through the surface of the tongue over the section. You've seen some different submucosal glands and other sections, but here we have a special organization of the mucosa. Okay, now it is the same section, but we went further through the same section, and we observe a papilla structure. Here, how do we understand the papilla structure? You see our epithelium showing the stratified squamous epithelium, and then the lamina propria and the other cellular elements of the lamina propria, the connective tissue elements, coming directly in contact with the basal layer, basement membrane, and they are forming a totally papilla structure, as I am showing you, pointing you at big papilla. And this papilla is very much in shape of a rectangular shape. So these type of papilla are named as the foliate papilla. As you know, as you remember, we have filiform papilla, fungiform papilla, and circumvallate papilla. But in that section that you have, we have the common papilla of the foliate papilla. And making another investigation through this papilla, we observed some special epithelium-bearing cells through the surface, and these are the taste buds. Taste buds are containing neuroepithelium type of cells, but they are still located in the stratified squamous epithelium, so they are special type of epithelium, but having the characteristic of neuronal induction. So these taste buds are responsible for the taste recognition. Okay, and these are the making a higher magnification of one taste bud. Yes, here you see the taste buds, individual taste buds. Located through the sulcus of the papilla. The sulcus of the papilla is a surface of the this characteristic of the foliar papilla on the lateral walls. In other papilla types, they can be on the upper surface as well. For example, in the circumvallar papilla or funiform papilla. But for the foliar ones, we always observe on the lateral walls. Okay, I'm going to the next section. Once more about the going through the muscle layer and submucosal layer. Okay. Now we can go to the next section. Now we are observing a compound, two compound organs actually. One of them having a more regular lumen, which is the trachea itself, the regular circular lumen, but the other organ is more, it has more imaginations, and it has a more many foldings. So this is the esophagus, and the esophagus lumen is still lined with a mucosa, and then we observe a thin lamina propria, and then a broad submucosa, and the next layer would be the tunica muscularis externa, and lastly there would be a serosa, or depending on the part of the esophagus that we obtain, some adventitia in the outermost layer. Once more, going back to the mucosal surface, epithelium surface, it is again a stratified squamous epithelium, and it is clearly visible the uppermost cellular nuclei more spindle shaped, and the basal cells more round cells. Okay, and then a thin layer of lamina propria and a broad layer of submucosa. Okay, and then the tunica muscularis, or muscularis externa we call, but going round the lumen epithelium, just neighboring to the mucosa, muscularis mucosa areas, some individual muscle cells were very near to the mucosal surface. They are muscularis mucosa, but here a wide area of muscle tissue. This is the muscularis externa. And remember, during the lesson we were talking about the part of the esophagus anatomically. The 1/3 of the upper part is more with the stratified muscle cells. The middle portion, a mixture of stratified and smooth muscles, but the lower part would be completely smooth muscle cells. Okay, so once you make a higher magnification, sometimes you can observe these straight muscle fibers. Actually, here more stratified muscle. So I can think that this is more on the upper layer. Okay, there would be still some sections of invading neuronal cells, plexuses, but they are more clearly visible in the next section of the stomach. So I'm going to the stomach preparation. This is dealing with the stomach. In the previous session, one of my a friend of yours, was asking, okay, sir, why is this stomach or why is this esophagus very small? I mean, is my esophagus so small? No, these preparations were made from laboratory animals. For example, those rodents, for example, rabbits or mouse or rats. Okay, so these are not human material. That's why the circumference is very small. Or you can still observe a big organ like this stomach present on your slides, on your microscopic slides. So just remember that we are still making our investigations on the vertebrates, but they are very common features, very similar anatomical and histological features. So we are still showing you those features on those animals. Okay, it is the same layer organization. First of all, the mucosa. Here, as you see, the mucosal surface, and then the lamina propria actually goes through the mucosa by very near neighboring with the gastric glands. It is still continuous with the lamina propria. But what about this broad area? This is submucosa, don't forget. So the submucosa is very thick or broad, and it contains more big blood vessels, maybe arterioles or venules. And then actually the thickest layer is coming, which would be the muscularis externa. Okay, so the muscularis externa has actually the circular and longitudinal orientation, but with the stomach we can sometimes have some diagonal orienting layer as well. So in some stomach sections you can still have almost 3 layers of muscle tissue, but it depends on the section. It depends on the 3-dimensional observation of this section. We are more interested in the mucosal surface. So going back to the mucosa. Okay. This mucosa is now with the epithelium type is different from the esophagus. So the first difference would be the epithelium type. Now, as you see, this lining epithelium is a different type of epithelium, which is the columnar type of epithelium. I would make another magnification. Now making a bigger magnification. Okay, clear. Now you can observe the tubular shape of epithelia, and as you see, those prismatic cells having showing more. Okay. Columnar type of nuclei showing us the a different type of epithelia. So until now we were observing a squamous stratified squamous epithelium, but now it's a columnar type of epithelia. How do we understand the columnar type? Here the nuclei are more spindle-shaped and they are just perpendicular to the lumen, and belonging to this shape, the cytoplasm is also almost rectangular. So columnar epithelium, the lining epithelium of the stomach, is making the uppermost lining of the gastric mucosa. And then this pits and the epithelium is continuous with the mucous neck cells. Mucous neck cells are forming the gastric pits orientation. So gastric pits are on the surface having the columnar epithelial and then the mucous neck cells. And the next layer, the mucous neck cells, still some of them here. And then the gastric glands. As you see, the gastric glands all through this section are present with basic cell types, which do we remember the basic cell types? One of them should be the more eosinophilic staining parietal cells. Here, more eosinophilic pinkish or reddish cells, the parietal cells. And going more deeper, the basophilic staining cells, more on the deeper portions, they will be the chief cells. Okay, we are responsible to recognize the chief cells and the parietal cells as well as the columnar epithelium. But please don't forget that there are also enteroendocrine cells, and you should remember that there are also some stem cells giving rise to all of these glands and the epithelium. So, but under the microscope, with these sections, we can easily recognize the chief cells and the parietal cells, as well as neck cells, mucous neck cells, and the columnar cells. Okay, another feature would be the more gross feature, which is here. Until now we were observing short pits but longer gastric glands. But now I'm going to another portion of this stomach, still on the same section. As you see, I'm going through all the mucosa. But now I am on a different part, having the gastric pits longer. As you see, gastric pits longer, but the gastric glands are shorter and narrower. So this new part is belonging to the pylorus section, pylorus portion of the stomach. But the previous view was about the cardiac region, the corpus region. So we can still understand our section with the basic information of the pits and the glands. And still, during the investigation, you can observe some lymphocyte infiltrations. Lymphocyte infiltrations are the presence of the lymph nodules, so they are present through the lamina propria, and some extent they can be found around the mucosa, but mostly in the lamina propria. Another lymphocyte infiltration here. Okay, and I'm more going around, and still having the thick muscularis externa. Okay, and we should have have another interesting things here. Okay. We are still in the muscularis externa. I'm going through the muscularis externa. I want to find an area between two muscle layers. Maybe you can understand what I'm looking for. Between the two muscle layers, the circular layer and the longitudinal layer, I'm trying to find the neuronal ganglion. Where is that? Dr. Zelia, excuse me. I'm trying to make a magnification. Ah, thank you. I am trying to show you some plexus, meandric plexus. Okay. We were observing these as you see, I'm in the muscularis externa. The muscle cells are smooth muscle cells are oriented circularly or longitudinally, but you see there are some different cells between those muscle layers. So it is very common feature of those neuronal cells with bigger nucleus, bigger nuclei, and even individual nucleoli. So this tip, at the tip of the arrow, you can see the nucleoli, nucleolus, okay? And still the bigger nucleus around. And the cytoplasm is stained also. So these cells are belonging to the meandric plexus ganglion, okay? So the muscularis externa of the stomach bearing those meandric plexus innervation is very critical for the functioning of the stomach. And you can see those meandric plexus in different more if you go through the section, but it is easy with their cytoplasm and nucleus, it's easy to recognize that. Okay, I'm going back for another section, which would be the tooth section, the last section for today. And usually we investigate the tooth in three portions, three levels. As you see, the crown of the tooth belonging to which tooth they have a different shape, but as you see, it may be an incisor tooth, so it's very sharp, and the crown. And then the neck portion would be the next part of the tooth. But since our tooth that we are investigating at the moment is an individual tooth, it is free of the bony alveolar structure, so it may be difficult to identify the neck section. But going down further, we are in the root portion, and the root portion is easily recognized by the pulp material inside. And as we go through this pulp material, you can see that axonal projections of the neurons invading the pulp here are very in this tooth are very much. So many axonal projections and many blood capillaries as well. So this part of the tooth is ending with the lowest part of the tooth, and the pulp material is surrounded by the dentin around, and the outermost layer would be the enamel. Okay, let me say, enamel material is the outermost layer, with giving some rod-like orient material, which, as you know, is the thickest material in our body. So the enamel material outermost, and then dentin, and the dentin is just neighboring to the pulp section. Okay, so this is the end up with the sections. I'm sure you can see more with your preparations, with your own slides. So please do not hesitate to work with those preparations, and please do not hesitate to ask questions about your sections. Good luck.